Okay, so it's my pleasure to introduce Dr. Brad Haubrich, who is a teaching postdoc in my lab. He arrived here, end of August, right? So he's been here a few months, getting settled. Some of you know him from Org Lab, right? He'll be doing Org 2 next semester, and Chem 1 in the winter, right? So Brad got his Bachelor of Science from Muhlenberg College in Pennsylvania, right? Then went on to do a PhD in chemistry at Texas Tech at Lubbock. And then what you'll be hearing about today is his work as a postdoctoral fellow at the Institute for Rare and Neglected Disease Drug Discovery. And the talk today is on slow binding screening for medicines of neglected diseases. And I'm going to maybe leave right here.
Thanks. It's great to be able to present some of my work from my last position. Today, I'd like to talk about. I'd like to start out with a brief introduction of. Can everybody hear me? A brief introduction of molecular mechanisms of action and what that means for drug discovery, including binding mode, and kinetics and enzyme competitiveness. Then I'll talk briefly about what a neglected disease is, and specifically human African Trypanosomiasis, which is the disease that I was interested in during this work. And then a specific enzyme target from parasite and a small molecule that we're excited about that can treat the disease. And if there's time, I have an example from a human target. And if I say anything that doesn't make sense to anybody, feel free to ask to stop me. I'd rather everybody understand what I'm saying. So my takeaway messages today are that molecular mechanisms of action can change a drug's efficacy, and that in testing compounds, doing the tests in such a fashion that you take into consideration molecular mechanisms of action is an invaluable tool for contemporary drug discovery. And that a simple method with these considerations for MMOA has identified a small molecule as a potential treatment for African Trypanosomiasis. And that these sorts of simple screens, particularly for slow binding, can be cost-effective strategies where resources are limited for drug discovery. So I'm starting off with a non-biological reaction. This is the oxidation of copper to cupric oxide. It's irreversible. It's naturally reason I can turn back to yellow. And when physical chemists talk about reactions, sometimes they draw diagrams like this, which are called reaction energy diagrams, where on the left side of the screen,
just choose one.
on the left side of the screen, I've drawn copper and oxygen at some resting energies, where the y-axis is energy, and on the right side of the screen I have cupric oxide at some other resting energy. And you'll note in this reaction that energy is released. There's lower resting energy of cupric oxide. And this term delta G is the energy that's either released or put into a reaction. And along the x-axis here, the x-axis is not time, the x-axis is placement of molecules in space. And there is a certain placement of molecules, the atoms, the placement of atoms in space. And there is a placement that's going to be higher energy, that's en route from copper and oxygen to cupric oxide. And that is, I have labeled as TS for transition state. And this occurs at a higher energy than the reactants. Perhaps an easier example would be burning a fuel. So obviously the burning of fuel, energy is released, but you need spark. You need that spark to get the reaction to go. So here I have a transition state drawn that's at a higher energy level than the reactants. And I chose hydrogen for this example because I wanted to demonstrate that a lot of reactions aren't quite so simple and have multiple transition states. And if I would have chosen some of the components of gasoline, there'd be a bunch of humps up there. But I just wanted to show that sometimes reactions are multi-step. But the activation energy is still proportional. Is still the difference in energy between the reactants and the highest energy transition state, regardless of where they're at. So compare that to a reversible reaction. Here I have bicarbonate in solution. So sodium bicarbonate is the active ingredient in baking soda, and it's represented by the middle species here. These are reversible reactions we're talking about. So bicarbonate in the solution can go to the left or the right on the screen at rates proportional to their delta G double daggers here. And likewise, the species on either side, on the right and the left, can come back towards the middle to bicarbonate. And at different rates of reaction. When equilibrium is established, you'll have species, all three species get a certain ratio proportional to their energies. But the system is dynamic, and at any given time you'll have the same ratio, but not necessarily the same molecules.
Right, that's cool.
Yeah.
You're saying basically the difference between reversible and irreversible in this description is where the normal environmental energy is either large enough in this case to kind of get you over the humps, so that way things kind of bounce around. But in the previous one, the drop was so big that the normal thermal stuff will not bring something back up the hill. Is that
Usually.
okay?
Usually that's the case. That's not always the case, but usually. In fact, well, that takes me into my next portion of the slide. There's another reaction associated with the dissolution of bicarbonate, and that's carbon dioxide coming off the solution, which is irreversible. Carbon dioxide leaves, and that's a much bigger energy. So consider a biological reaction. So this reaction I've drawn, we all perform this reaction. The molecule in the top left corner is called arachidonic acid, and we have proteins or enzymes called cyclooxygenases that catalyze the formation of molecules that look something like the right molecule. These are called prostaglandins. I'll call cyclooxygenases COX for short. Prostaglandins are hormones that are responsible for such physiological responses as vasodilation, platelet formation, sensitivity to pain, fever, and some other things. This reaction to prostaglandins doesn't occur without COX. So if you wanted to reduce somebody's fever or sensitivity to pain, for instance, you might make an inhibitor of COX, where I'm reacting some molecule with COX to make some inactivated COX. And if I made an inhibitor that had a reversible binding mode, I would establish some equilibrium. And at any given time, I would have a dynamic scenario where some of my COX was active and some of my COX had some inhibitor bound to it that was inactivated. And in this scenario, I could increase the dose where I would shift equilibrium that I had more inactivated COX. And when I'm talking about reversible enzyme reactions, enzymes and proteins are very large molecules, and the reversible reactions are typically an inhibitor occupying some space inside the protein that's not a covalent bond It's not chemically bound to the enzyme, but rather just occupying some space and making it not able to do what it ordinarily would do. In this case, make prostaglandins. So compare my reversible case to an irreversible case. In this one, I have two steps similar to the bicarbonate. So I'll have some sort of equilibrium in the first step that's reversible, but then I have an actual covalent bond forming, and the second step is irreversible. So if I had an inhibitor that was the lower case, the irreversible case, I might have a longer sort of an effect, and because I'm taking OX out. So, for instance, if I was worried about platelet formation is a very long biological process relative to some of these other things. So, an inhibitor for both of these might work for fever and pain, but wouldn't necessarily work for platelet formation. But my second guy takes COX out and may very well work for inhibiting platelet formation. And you can think of the top reaction being somewhat of a dial, more dose, more effect, and the bottom mechanism being like a switch-like behavior, where you're shutting off the COX. And you know these molecules is ibuprofen and aspirin, and this is why we don't feed children aspirin anymore, and also why patients at risk for thrombosis might be on a low-dose aspirin regimen, but wouldn't be on a low-dose ibuprofen regimen, because ibuprofen has this equilibrium and doesn't work at the same time. But there is another option, and you probably haven't heard of Indomethacin. It's by prescription only. Excuse me. But again, it's a two-step mechanism. But the second step, remember I said that most reversible reactions for enzymes will have just occupied space and not covalent binding. So my second step is that. My second step is just a change in the way the atoms are arranged, but it's not covalent and it's not irreversible. But it is a thermodynamic sink, much lower energy on the right side. So I'll have rates going for the first reaction proportional to that difference, and for the second proportional to that distance. But my irreversibility will be proportional to those energy differences in the reverse direction. But you'll notice the one I have drawn is very high and insurmountable, and this would it takes a very long time for the reverse reaction for the inhibitor to let go of the enzyme. And we call these sorts of behaviors slow binding, or pseudo-irreversible, or insurmountable. And the whole study of these binding modes is binding kinetics. So I won't bore you with a bunch of math. There are many different kinds of mechanisms. Sometimes there's one hump, sometimes there's two humps, sometimes there's more than that. But for the simple case, let's just take one-step mechanism that's reversible from enzyme plus inhibitor, E plus I, to EI, to an enzyme inhibitor complex, EI. That most pharmaceutical chemists will talk in terms of thermodynamic terms, and they're interested in a number called Ki, which is short for inhibition constant, and it's the ratio of how much of these I have to how much of these I have, but it's also the ratio of the rates in the reverse direction to the forward direction. And we call those rates, the rate constant for those K on and K off. And so we're interested in K off because it's very slow to unbind, and that's what we're looking for. Our target is a pathogen. And you can imagine you can imagine some scenarios for medicines where rapid equilibrium would be beneficial, such as fluoxetine is a serotonin receptor agonist. And if you had a switch-like behavior, you may very well turn your depressed patient into a manic patient, which isn't much better. Similarly, antihistamine receptors are often this rapid equilibrium type. But for infectious disease, which is what I'm working on, and this work represents infectious disease. Yeah, a protein or enzyme target from the pathogen, for bacteria, I have examples of bacterial and the HIV virus. These molecules are irreversible. And if you get switch-like behavior to pathogens, a pathogen target, hopefully you'll kill the pathogen and cure the disease. There are non-infectious disease examples of irreversible medicines, including those for, I have two examples for epilepsy and COPD. And there aren't many examples for pseudo-irreversible inhibitors, mostly because the people who are interested in binding kinetics up until about now, and even still now, have been interested in things like antihistamine receptors and other receptors that we have that you can't have too much of, or if you get a little bit longer of a residence time, you get a little bit better of the therapeutic effect. But you can imagine for pseudo-irreversibility, and again I have some non-pathogenic diseases, diabetes and COPD, for instance. But you can imagine, again, infectious disease would be beneficial to go in and turn off your whatever you're targeting. So acyclovir, for instance, is a medicine for chickenpox and other herpes viruses. And imatinib is a recent drug for leukemia and Philadelphia chromosome syndrome. You can imagine cancer's another obvious, besides pathogens. You want to go in and kill your cancer, shut off whatever you're targeting. And one last bit about mechanism of action that it's necessary to mention is inhibitor competition. So if I have an inhibitor that occupies some space in my protein or enzyme that's not where my normal substrates are. So for instance, if I have a COX inhibitor that's going somewhere else than where arachidonic acid goes, it doesn't matter if I have arachidonic acid or not, my inhibitor is going to work. And compare that to the bottom case, the competitive case. If my inhibitor goes where arachidonic acid is going, if I have arachidonic acid around, my inhibitor is not going to work that well. This is, and as a reminder, I'm looking for this. I'm looking for some change in the enzyme after my inhibitor is found that I create some thermodynamic sync and create kinetics that the reverse reaction doesn't occur. So what is a neglected disease? Neglected diseases are infectious diseases that affect impoverished populations. They typically happen in the tropics and in developing nations. So there aren't a lot of resources for research, and there's not a lot of market for pharma. So these diseases often go without advances in drug discovery for a while. And the World Health Organization lists about 19 of them at the moment, and to break down, there are viruses, mycobacteria, fungus, roundworms, flatworms, and tapeworms, and these three here are protozoan parasites. These particular protozoan parasites are related to each other, and not related to some other protozoan parasites that you may have heard of, the one that causes malaria. And so there's hope that with these in particular, because they're because they have a lot of the same characteristics, that if you find a treatment for one, that you may have found, fingers crossed, that treatment for one of the others. And I'm interested in human African trypanosomiasis. These are called kinetoplastids because they have a special organelle called the kinetoplast. But human African trypanosomiasis is caused by Trypanosoma brucei, which is the infection is deadly if it's left untreated. The parasite enters your bloodstream upon blood meal of the tsetse fly. And there are two subspecies of T. brucei that infect humans. And they occur in eastern and western Africa. And the diseases are called West African and East African trypanosomiasis. The tsetse fly is native to sub-Saharan Africa, so most of the cases are there. There are about 70 million people in these 36 countries at risk for the disease. There, I got a picture of a tsetse fly to the top left. 30,000 cases is actually pretty low, and that's down in the past several decades. And a lot of that is not due to the chemists, but due to vector control. And if you go to Lake Victoria, you'll see likely these blue tents, which are tsetse fly traps. So rather than spending the money on drug discovery, they try to get rid of the fly population. Bottom left is a picture of the parasite next to some red and white blood cells. The parasite is coated, one of the ways it skirts our immune system is it's coated in what's called a variable surface glycoprotein. And by the time your body starts making antibodies, the parasite has changed what it looks like on the outside. One of the challenges for pharmaceutical chemists is that in late stages of the disease, the parasite crosses from your bloodstream into your central nervous system. And we all have a what they call the blood-brain barrier, and getting drugs across the blood-brain barrier is an extra challenge for the chemists. And there are currently 5 drugs for sleeping sickness. One's still in clinical trials. The last one that passed clinical trial was approved in the 70s. There's resistance growing. So some of them don't, most of them don't cross the blood-brain barrier. All but one need to be administered by shot. And they all have pretty awful side effects. So there's need for new drugs. So my target in this work was a protein kinase. And protein kinases, this is a generic reaction that protein kinases catalyze. And they transfer a phosphate group from ATP, one of the biological molecules of energy, this phosphate group onto protein. And we have about 5,000 of these, 5,000 different kinds of these. And they have a myriad of functions, including cell cycle and apoptosis. So your body, for instance, your body a cell will get a signal that it's time to die and move on, make room for the next cell. And the signal will trigger this reaction where a protein kinase phosphorylates another protein kinase. And let's say my left one would be dormant. My non-phosphorylated protein is dormant and phosphorylated, and it's active. So then that protein kinase phosphorylates the next one, and so on, and it's a big chain reaction. And then your cell dies. When these are often dysregulated in cancer. So protein kinases are popular oncological targets, including imatinib, which I mentioned earlier, is a slow binding inhibitor. With imatinib, they didn't know that imatinib was slow binding until after it was approved and hit the market. And it wasn't until after that they discovered that it had slow binding characteristics. So I mentioned we have about 500 of these Trypanosome brucei has about 150, over 100 of which are essential. So, like I said, if you can target something that you need, that the parasite needs, and turn it off, you hopefully will kill the parasite. These 19 of the 150 are totally different than anything you and I have, or that scientists have found in other organisms. And then the others have varying degrees of similarity to ours. My target was called GSK3 beta, and it's about 35% similar to our GSK3 beta. And this is my reaction. I have a peptide called GSM that will get phosphorylated from ATP. But I'm not interested in that reaction. I'm interested in this reaction, the inhibitor reaction. And as a reminder, I'm looking for this slow binding characteristic of an inhibitor. So my GSK3 from T. brucei exhibits all the normal reactivities as I increase time or concentration of GSK3. I get a linear response and increased activity proportional to those. This is important. I had to play in this region. I want my reaction times to be 5 minutes. Sure, my reaction times have to be short because I'm trying to find long reaction times of my inhibitor. And if my enzymatic reaction is too long, I won't be able to tell the difference. Oh, and my substrates exhibit normal responses that you would expect as you increase substrate concentration. So on the left, I wasn't the first one to do T. brucei GSK. On the left was my control inhibitor. I was reported before I picked this project up that this compound, termed GW8510, is a potent inhibitor of Trypanosome brucei GSK. And on the x-axis is a logarithmic scale of concentration. And at some low concentration, I have zero effect on my enzyme. So any lower than that doesn't make a difference. And at some high concentration, I totally knock my enzyme out. It doesn't matter if you add more. So my gray line is an ordinary experiment. My black line, I've allowed this second reaction to occur a half an hour beforehand, before I run my enzymatic reaction. There's no difference. And the left side is looking at there's no difference between the gray and the black line. And this is because GW8510 is a rapid equilibrium. And allowing the two to interact for half an hour doesn't matter because they're popping in and off. And when I put my substrates in, the substrates pop in and off. And some of them production doesn't operate. GSK operates the way ordinarily. On the right side, this is the molecule we're excited about. It's termed Untitled. And you can see when I allow the inhibitor to react for half an hour beforehand, I actually increase the potency. And I need smaller amounts of titoglucin to get the same effect when I allow pre-incubation. Similarly, I don't have them drawn here, but you can check for competitiveness. Both of these inhibitors are competitive. By running the same experiment at high concentrations of substrate, they would both be shifted to the right. These are typical experiments for time dependence. They're usually done as an afterthought. They're also a lot of reagents, a lot of time, a lot of money. So we're interested in streamlining the process and having a sort of a litmus test of is my compound worth exploring if it's got slow binding or not. So we're advocating a four-point method where 2 of my points had this half-hour pre-incubation, 2 don't, and of those 2, 1 is at high and low concentrations of my substrate. And you can see, I flipped the one. Sorry, the right side now is my non-slow binding inhibitor, and my left side is Trypanosomiasis. And as I go down, going from top to bottom, I've increased ATP concentration. Most enzyme, most ATP inhibitors are competitive, which is troublesome because most cells have oodles and oodles of ATP. But you can see I kind of washed out my competitiveness with the pre-incubation, and increasing ATP with a slow binding inhibitor still works. Another experiment that says sometimes the slow binding people do, which is even more reagent and more time and more expensive. This is called the progress curve. So at the top you'll see I've increased concentrations of everything with my enzyme, and tested my reaction over the course of time. So I'm looking at what the effect of time is, and what the effect of higher, from green to blue to red is higher Trypanosomiasis. And you can see that at low concentrations of Trypanosomiasis, I'm co-incubating everything together now. There's no pre-incubation, I'm throwing everything together. And there's very little effect at short times. But as I increase either the reaction time or the concentration of Trypanosomiasis, I get an amplified effect. And whereas if I don't have slow binding, I have a linear effect. Doesn't matter how long I let the reaction go, it's always just going to be the same relative inhibition as the time was before. And you always like to see biological data. I have collaborators who grew the parasites, and the compound works in the parasites. Nice. So, plenty of time. My next example is from humans. So this article I have on the bottom of the screen came out just a couple weeks ago. Hit the mainstream media. If you were reading the news a couple weeks ago and you read about a miraculous molecule and/or a miraculous enzyme that's going to make us all younger, this is what they were talking about. So the enzyme is called nicotinamide mononucleotide adenylyl transferase, or NMNAT for short. And NMNAT takes NMN and ATP again to make NAD, one of the other biological molecules of energy. This enzyme reaction is reversible. We have three NMNATs, three different types of NMNATs that humans make. And they're expressed variably in different types of tissues. In particular, certain cancer types have either higher or lower NMNAT levels. So we're hopeful that maybe we have a new oncological target. So I'm looking again for slow binding inhibitors. Again, I ran all the right experiments. My enzyme behaves normally, and I get commensurate increases in activity with time or protein concentration. And the National Cancer Institute has large compound libraries. There's 140-ish approved oncology drugs, as well as 700-some drug inhibitors that will kill cancer cells in the petri dish, but aren't quite safe enough for human beings to take. So these 912 compounds I screened against NMNAT, and my results weren't too promising. I didn't get that many. I only got 35% inhibition, and what I would consider a pretty high concentration. And I removed metal organics, because metal organics are often toxic and often expensive. And I was left with 7 inhibitors, and of these 7 inhibitors, I passed these through a time dependent screen, and all 7 were time dependent. And only one was cheap enough for us to buy and pursue for subsequent experiments. This is NMNAT is of interest to some of the pathological people, or pathogenic people. So NMNAT in bacteria, for instance, there's a lot of research and some decent inhibitors. NMNAT for humans is pretty resilient, and for this particular NMNAT, the best inhibitor I found in the literature was not that great, and in fact it was about 4% of my, I needed 4% of the concentration that the other guys needed, if I use a pre-incubation. So I actually couldn't get my inhibitor concentrated enough if I didn't use a pre-incubation, because it wasn't that active. But if I allowed my inhibitor and my enzyme, NMNAT this time, to react beforehand, I got 50% marks in chemistry, molar amount. My 50% marks here are about 3/4 of a micromolar in the forward direction, and about 1/4 of a micromolar in the reverse direction. And the best one in the literature before this work was 10 micromolars to get the inhibition. And because my detection method for these experiments was a little different, and I was limited to some of the experiments I could do on the left side now, I varied my pre-incubation time. And you can see that as a function of reaction time of my inhibitor and my enzyme, I get more and more inhibition. And on the right side, rather than the 4-point I just showed you, I don't do high and low concentrations of substrate, but instead I pre-incubate with one substrate versus the other. So I pre-incubate, I get more inhibition, meaning I'm slow binding. I pre-incubate with one or the other substrates, and my enzyme gains some of that activity, and my inhibition goes down, meaning that I'm competitive for actually both substrates in this case. So, to reiterate, my takeaway messages are the molecular mechanisms of action are influence of drug efficacy, assay development with consideration to these ideas is an invaluable tool for drug discovery. And that a simple 4-point method is identified Trypanosomiasis as a T-Brucey GSK inhibitor. And that simple screens like these, in particular for slow binding, can be cost-effective strategies for testing potential medicines where resources are limited. And so I'd like to thank my collaborators, in particular my last advisor, Dr. David Swinging, and my buddy Jared LePatten, who made my little snail mascot. And with that, I thank you. Other questions? I hope. Okay.
So the 4-point method in terms of testing for the kind of time dynamics of these things. So you said that it's kind of more time effective and cost effective. Is it like what kind of difference do you think? Because at one point you kind of made a comment about how to do these plots is like a really long time.
Okay.
Yeah. So what kind of gains would you have in using this?
So my flow chart. Typically, pharmaceutical companies or nonprofits like our industry will run through thousands of compounds at one concentration, and see, and then they'll decide. In this case, 35%. And if I have more than 35% inhibition, I got it, and that's worth following up. Typically, people don't do the time-dependent thing until way after later in the process. But the difference between, so each of these data points is, these are 3 sets of duplicates. And then all the data points are over the spread of the concentration range. And I know from, so sometimes it's difficult because if you don't have these behaviors, like the one on the right, you do have these behaviors. You might not guess the right range.
Right.
So you have to run a longer, wider range of concentrations. Where if you run the gray one, and then you take that little, you take where your 50% mark is, and then just run your with or without perturbation, to avoid having to run a bunch of data points and a bunch of these experiments. So that,
Yeah, so you're talking, at least like a factor of 10 then, because I mean, you're going to have.
Typically these will have 8 to 10 data points times duplicates times 3 experiments. And then if you didn't guess right, if you are time dependent, then you have to redo the experiment and shift the data points. And if you're just doing this blindly, I mean, there are many hits. These aren't very good inhibitors, but we specifically were looking at kinase inhibitors. So there's a lot known about kinase inhibitors that didn't necessarily make the market. So we had good hits, but not all of them. And to spend the time and money on them. Are they or are they not time dependent? So we were looking for a test. Because certainly some of these experiments, in the progress curve, I can come up with all sorts of other mathematical constants and other things that are useful that I do. On the right side, I have other constants, and that's useful. But you want to run these types of experiments. Any other questions?
So what actually happens during the incubation time?
In this experiment?
Yeah.
So in this experiment, I load my substrates, enzyme, and inhibitor, and I run the reaction for either 5, 10, 20, or 30 minutes. And at low concentrations of inhibitor, at low times, I don't have enough time for Trypanosomiasis to bind properly. So the difference between like this point here, there's really no difference. So if that was what you were looking at in your screen, it doesn't look like it worked. It didn't work. Whereas over time, you start to get this thermodynamic sink of Trypanosomiasis and GSK, and yeah, the activity it's no longer a linear relationship, and as time goes on, you get more and more of that knockout effect. Does that make sense?
Well, that's it.
Thank Brad for